Hi guys, so today I'm going to tell you a little about section 2.3. So we just had a quiz about this, and it's mainly about carbohydrates, lipids, and yeah. Okay, so first, what we need to know is that we're going to talk about carbs. So carbs can be divided into three main groups. Do you know what those are? They are monosaccharides, disaccharides, and polysaccharides. So what is a monosaccharide? A monosaccharide is something that has one glucose, basically, or one of the it's the building block of a carbohydrate. So basically what we can go through here is glucose, as I mentioned, and then we can move on to fructose. Here you need to remember that there's 4, here there's 3, and here there is 3. These are the main ones. So you have glucose, fructose, galactose, and ribose. So glucose is found in most things, fructose is found in fruit, galactose is found in something, and ribose is found in your DNA. Okay, now disaccharides. We have maltose, which is glucose plus glucose, lactose, which is galactose plus glucose, and we have one more, which is sucrose, which I think is fructose and glucose, I believe. And then last, we have polysaccharides, or the most complex carbohydrates, which are going to be starch, glycogen, and cellulose. So we're going to go in a little bit of detail in these three. So these are basically the carbohydrates that you need to know. Okay, so starting off, let's talk a little bit about the first one, which is starch. So where is starch found? Starch is found in plants, right? So in plants, they'll use starch as a storage of energy. So they store their energy, which is carbohydrates, in the form of starch, which is a polysaccharide. Now, starch can go into two categories. It can go into amylose and amylopectin. Okay, so here's where it gets a little bit more complex. Amylose is the non-branched glucose alpha. So starch is glucose alpha only. So when you think of starch, you think of glucose alpha. Amylopectin is the branched. So basically in branch, it won't go straight like this. It'll go like, it's not exactly like that, but it's not straight. It's not linear. So that's amylopectin, it's branch, and it's also glucose alpha. And yeah, that's basically what you need to know about starch. Now, moving on to what's next, we have glycogen. So most likely, your teacher is going to ask you to name some common similarities and differences between the carbohydrates. So glycogen, we can start to see some similarities to starch in the way that glycogen is a stored energy, but for humans and animals. So where is starch found? In plants. But glycogen is found in humans and animals. So that's difference number 1. So keep these in mind. Glycogen. So glycogen is branched. Another similarity, which is amylopectin, also branched. And it is also made out of glucose alpha. Did I get that right? Yes, alpha. That's a similarity. Glycogen is stored in muscles. Oh, I did that right. Muscles and in the liver. So these are for energy when you need them. Now, last polysaccharide, which is cellulose. Oh, and I forgot to add on that glycogen is soluble. Oh, soluble in water. Yes. Okay, cellulose. Cellulose is, have you ever heard of the xylem in a plant? Do that in green. Xylem of a plant is where the water moves through. So it needs to be able to have adhesion with the water so the water can stick to the side to be able to climb up. So when the water is climbing up, it needs something that's not soluble, or else it would dissolve. So the first property of cellulose, it is not soluble in water. There might be an A in there. So it's not soluble in water. That's what you need to know. Also, what's special about it, it's made up of glucose. Is it alpha? No, it's not. It's glucose beta. So we're already seeing differences here between the cellulose and the starch and glycogen, which are glucose alpha, and this is glucose beta. Okay, that is what you need to know about cellulose. You also need to know that it is not branched. Cellulose is straight, and that's very important for you to know. And okay, now that we've got those carbohydrates over and done with, we can move on to condensation. And what's the opposite of condensation? Hydrolysis. Do you know what those are? Well, we're gonna find out. So condensation is when you're building. It's an anabolic reaction. Condensation is a type of anabolic reaction that will use energy to build. So you're going from mono to di to poly, right? So you're going from, let's say, a glucose to a sucrose to a polysaccharide. Okay, so condensation. Let's take this. Whoa, that is not good. Let's take this glucose molecule. Okay. Yes. So this glucose molecule, it's going to be a glucose beta molecule. And okay. So first you got to put in our H's. Here there's an H. This is going to be an O. The OH is up, H is down, H is up, OH is down, H is down, OH is up, OH is down, H is up. Okay? So this is our glucose molecule, glucose beta. Now, when we want to perform hydrolysis, we need not one, but two glucoses. Let me put these in the middle so we can work on them. My goodness. Okay. Hydrolysis. Now, what's going to happen in hydrolysis is that this reaction is going to... So condensation is going to be in... Oh, I meant condensation. Okay, so condensation... Let me repeat that part. Okay, stop. Okay, so condensation is going to release water as a byproduct. So in addition to, let's get right, condensation is building. So when we're performing condensation, the O and the H will bond together, will come together basically, and these will form, can you guess what? One of the byproducts. Building, and byproduct is H2O. So what's going to happen is these are going to turn into H2O. But wait, there's two O's. And we're using two H's, so these are gone. We're only using one of the O's. What happens to the other one? Well, in fact, what will happen is that these two glucose molecules will form something sort of like a bond. Actually, it is a bond. but it's going to be called a special type of bond. I'm going to erase these because they're gone. So this leftover O from over here, it's going to come take a spot right here, and it's going to be able to connect to both of these. So basically, what just happened is I connected these two glucose molecules by this O bond. Now, what is this O bond called, you may ask? It's called a glycosidic bond. Okay, so these two glucose molecules were bonded by a glycosidic bond through a condensation reaction to form maltose plus H2O as a byproduct. That is very important. It's a key idea. So, two glucose molecules perform the condensation reaction to result in one maltose plus one water molecule. That's basically the gist of what just happened. Now, hydrolysis. What is hydrolysis? Hydrolysis is the opposite of condensation. So, we said that condensation releases water and builds two. So, what is the opposite of that? Hydrolysis, which breaks them apart, breaks two things apart, and it, instead of releasing water, it absorbs water, and it's a type of catabolic reaction. Okay, so how do we go from this figure to that figure in hydrolysis? Well, I am going to tell you how once I move this little molecule up a little bit. Yeah, okay. So, from here to here, it's condensation. But if I go the opposite direction, if I'm going from this bonded maltose, so I'm going from maltose to glucose, that, and adding water to the maltose to get two glucose molecules, that, my friends, would be hydrolysis. If you have any questions, feel free to comment. And yeah, thanks for listening.